Alright, so your wonderful teacher bought a brand new pad to try and write on this. I'm not using the picture of me so that you can actually see this. Now you'll notice I have up here anaerobic respiration, also known as glycolysis. I want to briefly walk you through some of these steps. Hopefully this is better. You can see it, the whole works. Before I start, don't freak out over all these names. That's not necessary. What you're supposed to know for the AP exam is the big picture. So I wanted to go through this, and I tried to find something as close to the green board that we used for those of you that already had in biology. But if you'll just watch the parts that are important, we're going to talk about. Okay, so what the deal is right here, what I have is a 6-carbon molecule called glucose. Now literally what the deal is, this I'm drawing right here is the cell membrane. Okay, I'm going to put CM. Alright, when it crosses in here, immediately we are going to use, and I'm going to put a minus, let me get a different color here. We're going to put a minus there. Alright, and we're going to put a minus there. We're going to use 2 ATP to flip-flop this thing around. You see all these. We're going to flip-flop this thing around. It is still a 6-carbon molecule, but the deal is that you're going to split it down into 2 3-carbon molecules. And by adding on these phosphate groups right here, what you're doing is not allowing that glucose to get back out through the cell membrane. It's trapped inside. So the whole point of this is to prepare glucose, break it down into 2 3-carbon molecules, and then what we'll see is that we're going to use those to do something else. So we have to use, I'm putting a minus, 2 ATP. I'm learning, so bear with me, 2 ATP. Okay? So what we're also seeing here is the pathway right here. Let me get some black. Alright, this pathway right here is literally going to go on down this side, and it would do the same right here. It's just that it's a pain in the butt to show both of them at the same time. Okay, so let's go back. We have glucose moves into the cell, and we're going to use now 2 ATP to break it down into 2 3-carbon molecules. At this point, we still have to prepare it for for it to be able to go on down the pathway. And remember, this is a biochemical pathway. So what we're going to do is we're going to have on each side an NAD go in, grab some hydrogen, and it's going to take that hydrogen out, which changes this molecule. Notice it's still 3-carbon. Then we're also going to have ADP go in and grab those phosphate molecules off, and we're going to literally do this twice. So if we're taking into account both sides, we are going to generate plus 4 ATP. Now notice up here it was minus 2. Down here it's plus 4. Okay? We still have 2 3-carbon molecules. Now this one's called pyruvic acid, and that's a one that you probably would want to remember. So we have 2 of these. Now, if you are a little yeast cell, and we'll try writing that. Let's say yeast. That's enough ATP for you to be able to handle it. It's a single-celled molecule. ATP, that's enough energy for me to do whatever I've got to do. If that stops there with yeast, it's going to stop here. We are going to make ethyl alcohol. If you are a little bacteria cell, that's enough for me, 2 ATP. But I am not going to make ethyl alcohol. I'm going to make lactic acid. This is what anaerobic respiration is all about. In other words, these 2 things right here don't need any oxygen, so it's called anaerobic. So what the deal is that we put a little sugar in with our yeast, give it some warm water, start letting it feed on some sugar and stuff, and it's going to take the glucose and it's going to turn it into ethyl alcohol and carbon dioxide. The ethyl alcohol is the smell you get from bread when you're making dough, or you actually take yeast and make wine or liquor or other things. On the bacteria, what happens is that, let's say, for example, you want to make sour cream, the bacteria will feed on the milk sugar and turn it into lactic acid. These are the end products, and that's all they're going to do. That's all they need is 2-ATP. But what about you? What are you going to do? Why do you have to go through this? And if you think about it evolutionarily speaking, of course, that's kind of where we all become similar. But anyway, so we're going to take our glucose. If we get to this point and there's no oxygen, your body is going to turn it into lactic acid also. Now, this is the muscle cramps and stuff that will build when lactic acid builds up in the muscles. And that's a problem, because what that's happening is you don't have enough oxygen to take it on the pathway. And so literally what your body has to do, you rub it, you start sending blood to it, it gets red, it cramps and starts moving that muscle around a bunch. Oxygen gets to it, and the oxygen will allow that lactic acid to turn back into pyruvic acid. And we can continue on the pathway to, say, for example, the Krebs cycle. So, brief overview. What the deal is. Glucose comes in, across the cell membrane, and insulin, of course, is what allows it to go in. We've already talked about that in cell communication. And as it comes in, we have to take it through a series of steps to keep it trapped and break it down. And so we use ATP, and then we're going to take and break it down some more, but we're going to make ATP. So we made 4 ATP. We also made the 2 NADHs. Okay? So keep that in mind. You'll need to know that later. So at this point, anaerobic respiration or glycolysis, if it stops here, that's enough for smaller organisms. But if we have oxygen present in larger organisms, it is going to continue on down the pathway. This is glycolysis. It is part 1. I'm adding just a little bit more on. I also want to comment that, let's say, for example, you aren't eating and you're not getting glucose, and you've used up all the glycogen in your liver and turned it into all the glucose, and so you're done. Literally, in different spots in this pathway. And if you ever take biochemistry, you're going to see like these great big long lines that come down through here. You're going to be able to use fats and proteins, which is why you had to study all that stuff. So there's proteins. Fats and proteins can enter down in here, and then they can literally be turned into some of these molecules in order for some of these things to occur. You won't get quite the bang for your buck, so it's not the best way, of course, to get energy. You'll find out in later pathways that you don't get the same amount of NADH or the same amount of ATP being made. However, as a fail-safe, and you don't have food coming in, this also will help you out a little bit. Comment, I forgot to make one more thing I forgot to mention. So our total down here of ATP that we made to the good is +2. So keep that in mind. We had -2 up here, and then we have +4 down here. That gives me ATP +2 ATP to the good. Now I'm done. We'll go on to the second part.